Hello students, in this module I'll be talking about the pollen-pistil interaction part 2, right? So in the previous module you saw that the pollen grain landed on the stigma and it started to germinate after the stigma had recognized it to be the right kind of pollen. Now the next step would be basically for that pollen tube to go down to the ovary, right, and enter the ovule through the micropyle. Is it okay? That'll be the next step. Right? So you must know that the pollen tube will now go down and try to enter the ovule through the micropyle. Yes, that's the pollen tube right there. Now remember, it is the filiform apparatus which is present in the synergids, and one of the synergids will help in the entry of the male gametes into the embryo sac by bursting of the pollen tube. Does that make sense? So now both the male gametes have entered the embryo sac. Even these events are part of the pollen-pistil interaction. Is that okay? So it is not just the recognition of the pollen and germination of pollen, it is also the entry of the pollen all the way into the ovule as well as release of the male gametes in the embryo sac, all constitute the part of the pollen-pistil interaction. Is it okay? Now let us see an example right behind my back. What I've shown you is a blown up image of 3 different type of pollen grains on the stigma, right? Right, on the top you see a yellow pollen. I'm going to call that at pollen number 1. Right above my head you see a purplish pollen. I'm going to call that as pollen number 2. And then again right above me on the right you see a spiny pollen. I'm going to call that as pollen number 3. Now these 3 different types of pollen has landed on the same stigma. But look at the center pollen, the pollen number 2. What do you see? You see that long germinated pollen tube. Is it okay? Which means the stigma knows that pollen number 1 and pollen number 3 are the wrong pollens. And pollen number 2 is the right pollen. And that is why it allowed its germination. Does that make sense? Now why would this happen? They say that the pistil has the ability to recognize the pollen based on certain chemical present in the pollen grain, which interacts with those of the pistil. And that is why it selects the right pollen and then allows the germination of only the right pollen. And the wrong pollens will ultimately just be discarded. Is that okay? Now remember that there are these botanists who have identified components of the pollen and the pistil, and then they have further said that why a pollen and a pistil recognition really happens, right? Or why this interaction really happens. Yes, and they have also described that after the pistil has recognized the pollen, then what are the events that lead to its acceptance or its rejection? All of this has been identified by the botanists very recently. Now this knowledge is going to actually help the plant breeders in the future to be able to manipulate the pollen and pistil interaction, especially when there are incompatible pollinations, right? And why would they do that? They would do that to produce these beautiful hybrids. Is that okay? So if botanists and plant breeders have to produce some desirable hybrids, then they have to have the knowledge of pollen-pistil interaction so they can manipulate things, right? So that even incompatible pollinations can be made successful and we can get these desired hybrids as per our choice. Is that okay? So in this module, I've talked about the second part of the pollen-pistil interaction. In the next module, I'll be describing the process of double fertilization in angiosperms. Till then, thank you and take care.